Class 11th, Subject Biology, Chapter 8, Cell the Unit of Life, Part 5. Good morning, students. Today we are going to study the structure and functions of endoplasmic reticulum, Golgi apparatus, and lysosomes. So the endoplasmic reticulum is a complex network of tubular membranes exclusively present in the cytoplasm of eukaryotic cell. The endoplasmic reticulum, in short ER, is a large organelle made of membranous sheets and tubules that begin near the nucleus and extend across the cell. The endoplasmic reticulum is a name derived from the fact that in the light microscope it looks like a net in the cytoplasm. This organelle acts as an important regulator of cell function because it interacts closely with a number of other organelles. Products of the endoplasmic reticulum often travel to the Golgi body for packaging and additional processing before being secreted. The endoplasmic reticulum can either be smooth or rough. Smooth endoplasmic reticulum is generally known as SER in short, and rough endoplasmic reticulum it is known as RER. And in general, its function is to produce protein for the rest of the cell to function. The rough endoplasmic reticulum, as the term indicates, it is rough because large number of ribosomes are attached to the surface. The ribosomes' functions is to make the protein. Sometimes when those proteins are made improperly, the proteins stay within the endoplasmic reticulum. So the endoplasmic reticulum is an organelle that's really a workhouse in producing proteins and substances needed by the rest of the cell. Typically, the smooth endoplasmic reticulum is a tubular network and the rough endoplasmic reticulum is a series of flattened sacs. So the main difference between SCR and RER is that RER contain large number of ribosomes, but on SER these ribosomes are absent. And the main functions of endoplasmic reticulum are, it is responsible for the production of not only production and secretion of steroid hormones. It is also responsible for the synthesis of essential lipids such as phospholipid and cholesterol. It is responsible for the metabolism of carbohydrates. Endoplasmic reticulum releases calcium ions which are necessary for the nervous system and muscular system. Then next is cell organelle, Golgi apparatus. It is also known as Golgi complex or Golgi body. These are membrane-bound organelle of eukaryotic cells that is made up of a series of flattened stacked pouches called cisternae. The Golgi apparatus is responsible for transporting, modifying and packaging proteins and lipids into vesicles for delivery to targeted destinations. It is located in the cytoplasm near to the endoplasmic reticulum and near the nucleus. While many types of cells contain only one or several Golgi apparatus, plant cell can contain hundreds. Depending on the type of cells, there can be just a few complexes or there can be hundreds. Cells that specialize in secreting various substances typically have a high number of Golgi. In eukaryotic cell, the Golgi apparatus is a manufacturing and shipping center of the cell. Under electron microscope, the Golgi apparatus is seen to be composed of stalks of flattened structure that contain numerous vesicles containing secretory granules. Typically, Golgi apparatus appears as a complex array of interconnecting tubules, vesicles and cisternae. The Golgi apparatus is very polar. Membranes at one end of the stalk differ in both composition and in thickness from those of the other end. One end, cis phase, acts as the receiving department while the other, trans phase, acts as the shipping department. The cis phase is closely associated with the endoplasmic reticulum. Golgi apparatus was discovered in the year 1898 by an Italian biologist, Camillo Golgi. And the functions. The Golgi apparatus modifies proteins and lipids that is received from the endoplasmic reticulum. These biochemicals leave the Golgi by exocytosis before being delivered to the different intracellular or extracellular targets. Protein processing. Carbohydrate regions of glycoproteins are altered by addition, removal, or modification of carbohydrates. Lipid processing adds phosphate groups and glycoproteins to lipids from the endoplasmic reticulum, such as cholesterol, to create phospholipids that make up the cell membrane. A lysosome is a membrane-bound cell organelle that contains digestive enzymes, and it is a subcellular organelle that is found in nearly all types of eukaryotic cells, and that is responsible for the digestion of macromolecules, old cell parts, and microorganisms. Lysosomes were discovered by a Belgian cytologist, Christian René de Douai, in 1950s. The word lysosome is made up of two words. Lysis means breakdown and soma meaning body. Lysosomes occur freely in the cytoplasm in animals found in almost all cells except in the RBCs. Lysosomes are called the suicidal bags of the cell. They contain hydrolytic enzymes. If the cell is damaged, the lysosomes undergo autolysis and burst open. This results in the release of those hydrolytic enzymes. The released enzymes then digest their own cell and ultimately the cell dies. Hence they are called the suicidal bags of the cell. Lysosomes originate by budding off from the membrane of trans-Golgi network, a region of the Golgi complex, responsible for sorting newly synthesized protein. Lysosomes serve two main functions. Number one, intracellular digestion. Means to digest food, the lysosome membrane fuse with the membrane of food vacuole and secrete the enzymes inside. The digested food then diffuses through the vacuole membrane and enters the cell to be used for energy and growth. Number two, autolytic action. Cell organelles that need to be get ridden and covered by vesicles or vacuoles by the process of autophagy to form autophagosomes. The autophagosome is then destroyed by the action of lysosomal enzymes.